Our next speaker is Luke Gandolfo, and he is currently a research officer from Terry Speed Lab in Walter and Eliza Hall Institute in Melbourne, in Australia. And Luke is the author of the paper of relative log expression plots. The RLE plots method is also used as the plot RLE function in the very famous scatter bioconductor package. So let's welcome Luke to introduce something for the RLE plots.
Thank you. Thanks for that introduction. And thanks also to the BioC Asia Committee for the opportunity to speak today. And thanks for everyone for coming along. So I'll be talking about RLE plots. And I should say that this is joint work with Ramya Malania, also from the WeHi. So I'm just going to minimize that. Okay, so the context for today's talk is visualization. And in particular for today, it'll be visualization of high-dimensional gene expression data. For example, from microarrays or RNA-seq or the NanoString platform. But actually, what I have to say will also have a relevance for other kinds of high-dimensional data like ATAC-seq or ChIP-seq or metabolomic data, etc. And just a footnote, the ideas I'll be discussing today are pretty simple. There's no sort of R package involved, although as mentioned earlier, there are R packages that do implement this idea. But nonetheless, I thought this would have wide interest for the BioC community because visualization is important to us all. So let's make a start. So why do we visualize data? Well, of course, one reason is just simply to see if there's any technical variation in our data. That is, differences in expression measurements between samples, which are caused not by biological differences, but by technical differences. And of course, a classic example of such technical variation is the so-called batch effect, associated with samples being prepared in different locations or on different dates or with different protocols, etc. And more generally, we're interested in visualizing just any kind of technical variation, whether we know its cause or not, because we care about seeing whether we've got technical variation in our data because, of course, it can have consequences. At worst, at best, actually, it simply adds noise to our analysis, and that can lead to missed discoveries. At worst, if there's any level of confounding between the biological conditions of interest and the factors that are causing the technical variation, something we're going to discuss later, then this, of course, can lead to false discoveries. So if we see technical variation in our data, we'll want to normalize the data. That is, adjust for or remove the technical differences between the samples and roughly leaving only the biological differences. After all, that's the thing we're interested in. And then, of course, we'll want to check whether the normalization we've applied has actually succeeded in dealing with the technical variability, although sometimes this is overlooked. And with this, I just want to make a note that there are sort of two different modes we can be in when we're visualizing data. One is simply just detecting technical variation if it's there. And then another mode we can be in is checking the success of a normalization we might apply to deal with that technical variation. And we need visualization tools for both these tasks. And of course, it's no exaggeration to say that the standard tool we use for visualizing our data is PCA plots. And for an audience like this, not a lot of explanation is needed, but I should quickly move on to say that I love PCA plots and I wouldn't give them up for the world. But what I have to say today is that there are situations where these plots are poor tools for detecting technical variation. So to explain what these situations are, let's look at some examples. So the first example is what I call the textbook data, because it comes from a wonderful textbook by Love and Irizarry, and it's microarray data. And it has 3 different biological conditions, A, B, and C. Doesn't matter what they are for our purposes. And as you can see, when we make a PCA plot of the data and color it by the biological conditions, we see biology. We see a nice separation of the biology along the first principal component. But it turns out that these samples were actually processed over 5 years. And the year of processing is an obvious source of technical variation here. In other words, a batch variable. Now, with that in mind, observe the following table. Note that almost all the Bs were processed in 02, 03, and 04. Almost all the As were processed in 05, and almost all the Cs were processed in 06. In other words, what we're seeing here is a very significant level of confounding between the biological condition of interest and the batch variable, in this case, year of processing. Okay, so let's make 2 PCA plots, one colored by biology and another colored by the batch variable, in this case, year of processing. So hopefully now you can start to see what the problem might be, because the question is, are we seeing batch or biological effects in these PCA plots? In other words, what's driving the separation of the samples along the first principal component? Is it biological effects as seen here, or is it batch effects as seen here? Given the high level of confounding in this data, it's very hard to tell from the PCA plot. And of course, you might be thinking, yeah, come on, this is from a textbook. Of course, they're going to show you a very, very bad example of what I'm trying to point out here. But here's an example that just came up recently, randomly in my own research. It's RNA-seq data on T cells. And when we do a PCA plot of the data, one colored by biology and one colored by batch, in this case, 2 different processing labs, again, we see a high level of confounding. These 2 brown samples, these 2 purple samples, these 2 bright green samples, and these 2 gray samples, they are all sitting in the purple group, purple batch over here. And all the other samples are in the orange batch. And again, the question is, are we seeing batch or biological effects here? Given the very high level of confounding, it's very hard to tell from the PCA plot. And I'll just note that we're visualizing log CPM here, so library sizes are being taken into account. So hopefully you can see what the problem is now. PCA plots are sensitive to both technical and biological effects. So if there's a high level of confounding between the biological conditions of interest and the factors that might be driving any technical variation, it can be hard to untangle their effects in a PCA plot. And the same point applies to other visualization tools, for example, multidimensional scaling plots, just as one example, that are both sensitive to technical and biological effects. And if this is going to be a problem for merely just detecting whether there's technical variation in our data, it's then going to be a problem for checking whether we've successfully normalized the data and dealt with the technical variation we've seen. So the conclusion here is that if we want to get to the bottom of this problem of confounding, we need a visualization tool which is sensitive to technical effects, but insensitive to biological effects. So a possible solution here is to use relative log expression, or RLE plots. And just a quick review for us, our gene expression data is essentially going to consist of a big data matrix Y, for example, like this guy. And we've got genes in rows and samples in columns. In this case, we've got two groups. And the entries Yij are going to be the log expression for gene I in sample J, where what I mean by expression here is intensity in the case of microarrays, or CPM in the case of count sequencing data, etc. So with that in mind, what is an RLE plot? Well, we take our big data matrix Y, and then we take each row and we subtract the row median across the median across that row from the row. Okay, take each row, subtract the median across the row. And then we end up with a sort of centered data matrix, or be it centered on the median. And once we've got our centered data matrix, we take each column and dump it into a box plot. So before I show you some examples, I might just linger on what's going on here. How do they work? Well, on a log scale, the measurements for each gene essentially consist of the following. We have a gene mean plus a biological effect, if any, plus a technical effect, if any, plus some variation, or mu-i plus dij plus tij plus epsilon ij. Now, if you make an assumption that the majority of the genes are not differentially expressed, then if there's no technical effects present in the data, the measurements for a majority of the genes are going to consist of mu-i plus epsilon ij, because we're assuming for this majority of genes, there's no biological effect, and we're assuming, for the sake of this argument, that there's no technical effects. So we'll have mu-i plus epsilon ij. Now, since the median across a row is essentially the gene mean, when we subtract that median, what we're essentially going to end up with is epsilon ij. So an RLE plot would, roughly speaking, essentially display epsilon ij's. That is, the box plots would be centered on 0, and they would be roughly the same size. So to sort of flip this argument around, if the box plots are not centered on 0, are not the same size, then this is a strong indication of the presence of technical effects. So to get a bit more of an idea of what's going on here, here's some cooked up examples. And here is an RLE plot that's, that looks good. It's showing that, it's indicating that there are no technical effects. And that is the boxes, the medians of the boxes are centered on 0, and the boxes are roughly the same size. And on the right is a very bad RLE plot where we don't have the median centered on 0 and the boxes are all over the place. That is an RLE plot that's strongly indicating that we've got serious technical effects here. Now, the claim I want to make is that RLE plots are sensitive to technical effects, but insensitive, or at least highly robust, to biological effects. And the evidence for this is chiefly going to come from a simulation study, where we're going to simulate data according to this model, where the gene means, the technical effects, and the variation will have these particular distributions. And all of this is pretty standard. We can talk about it in question time if you want, but it's the sort of stuff that underpins a method like limma. So the other thing that's going to be important here is the differential expression, the dij term, but I'm going to explain that later. Now, using reasonable parameter values, we'll simulate 10,000 genes with 3 groups, each with 3 samples in each group. And if there's going to be a batch effect, I'm going to make it so that it cuts the samples into 2 batches, and incidentally will completely confound 2 of the groups. Then I'm going to make an RLE plot. So here are the results when we have no differential expression in the data, no biological effects. So if there's no batch effect, then the RLE plot looks as it should. The boxes are centered on 0 and they're roughly the same size. And if we insert a small batch effect, cut in the samples into 2 batches, these samples in one batch and these samples in another batch, we see the batch effect clearly stands out in the RLE plot. So this suggests the RLE plots are sensitive to technical effects. Well, great. So at least we're in business on that front. But the real question is, what happens when we inject a large amount of differential expression? And for our purposes, we're going to proceed very simply here. We're just going to take a single large value and add it for every gene we're going to deem to be differentially expressed. So we're going to take our Dij term to be, for example, just 5, which, because we're on a log scale, represents a 32-fold change. That's a serious piece of biology. But what's more is we're going to make 2,000 genes differentially expressed for each group relative to the other 2 groups. So in other words, overall, we're going to have 6,000 genes differentially expressed, and they're going to be all differentially expressed to a very large degree. And moreover, we're going to make things even more interesting by making the scenario unbalanced. We're going to make 90% of the genes be up and 10% be down. And when we do this, when there's no batch effect, the RLE plot looks as it should. They're centered on 0 and roughly the same size, but which indicates that it's basically ignoring the large amount of biology that's in the mix here. But when we insert a small batch effect again, it stands out like a sore thumb, despite the fact that there's a huge amount of biology here. So it seems that RLE plots are insensitive to biological effects, but still sensitive to batch effects in the presence of a large amount of biology. And is this a solution to the problem of confounding, where we needed a visualization tool that was sensitive to technical effects, but insensitive to biological effects? Well, in the case of the textbook data, the question was, are we seeing batch or biological effects here? Well, an RLE plot shows that we're not seeing biological effects, we're seeing strong batch effects, because these tend to be below 0, these tend to be above 0, and we also see a large amount of within batch variation too, showing that it's not just the batch effect, it's not just a batch variable that's driving technical variation here. What about the T-cell data? Again, are we seeing batch or biological effects? Well, the RLE plot shows that we're seeing a serious batch effect. And again, we note the within batch variation. So at this point, you might be wondering, well, why don't we just use simple box plots, as some people typically do, which is to take each column of our data matrix and just dump it into a box plot. Of course, the idea being that if the boxes aren't lined up, this suggests the presence of technical variation. Well, the problem with box plots are that they're not particularly sensitive to technical variation. And this can lead to missing our ability to see technical variation if it's there. So for example, if we're just merely trying to detect technical variation, for the textbook data, when we did an RLE plot, we saw significant technical variability. But had we done box plots, things look pretty good. Things look pretty nice. So in this case, RLE plots are more sensitive, and so it allows us to better see the technical variation here. What about an example where we're trying to assess the success of a normalization? And for this, let's take a particularly bad data set where we've got significant batch effect for 3 different labs, although I might point out that the biology is reasonably well distributed here. And let's ask, and so this is a particularly bad data set. And indeed, you don't need an RLE plot to see problems here. The box plots show the batch effect just fine. So we definitely need to normalize the data here. And with the data set this bad, we need to check if we've succeeded. Well, why don't we try quantile normalization? Well, if we do box plots after quantile normalization, it looks, well, perfect, right? But have we perfectly normalized the data here? An RLE plot shows that we certainly haven't. There's still a significant batch effects here. And in case you're wondering what's going on here, why does this look perfect? We can chat about that in question time. What about regressing? What about something simple like regressing out the first principal component? After all, that was capturing the batch effect, and the biology was reasonably well distributed between the batches. So it seems like it might be a reasonable thing to do. And indeed, the box plots look pretty good after we regress out the first PC. But the RLE plot shows that we've still got quite a way to go in removing the technical variability here. So the thing is, we see the same sort of effect for other normalization methods like SVA or RUV. And I'm not saying that these methods can't remove technical effects in this data. Of course they can. You might just need to regress out more principal components, for example. My point is that you don't need to do much to get good looking box plots, while the RLE plot can tell another story because it's more sensitive. So since RLE plots are more sensitive, they put us in a better position to check the success of the normalization. And you might think, what about PCA here? Well, indeed, it is relevant here, but the point is, as I said earlier, if there's any significant confounding of your biology and batch, maybe unbeknownst to you, then PCA isn't particularly useful. So you're still going to want to use a tool that's sensitive to technical effects, but insensitive to biological effects to assess your normalization. And I'll just race through a couple of other advantages. RLE plots provide a clearer yardstick for normalization success, namely that the box plots be centered on 0 and have similar widths, because as you can see here, the PCA looks great. It looks like the normalization succeeded. On the other hand, the RLE tells a different story. RLE can sometimes tell the difference between different normalizations when PCA can't. So here's some nanostring data and 2 different normalizations applied to it, A and B, both look great, and indeed both look equally good according to the PCA. So the question is, which is better? Well, an RLE plot reveals that B is much, much better than A. And indeed, we know B is much better than A for a host of other reasons, and I'll refer you to this paper for those other reasons. And the last point is, RLE plots don't need color. And that's particularly useful when you just don't know any of the technical factors that might be producing unwanted technical variation. Because if you don't know any factors like that, you've got nothing to color your PCA. But an RLE plot doesn't rely on color. It has a different criteria. And that's particularly important when you don't even know your biology. So you can't even color your PCA plot to use that as a measure of whether you've got technical variability in your data. So to conclude, we're advocating that you use PCA. You don't give up using PCA because we love PCA, but you just supplement the use of PCA with RLE plots. And you want to do that because, because RLE plots don't see biology, they're not going to tell you things like whether you've removed biology when you've normalized your data. And of course, you shouldn't rely just on data visualizations to assess the success of a normalization. There's a host of other things there that you should do. So in summary, common visualization tools, for instance, PCA, are poorly suited to detecting technical variability and checking normalization success in situations involving significant confounding, whether they be known or unknown. RLE plots are more sensitive to technical effects, but they're sensitive to technical effects, but insensitive or highly robust to biological effects. This makes them useful for visualizing technical variation in situations involving confounding. RLE plots are more sensitive than box plots. This makes them a better tool for detecting technical variation and checking normalization success. RLE plots can provide a clearer yardstick for normalization success, can tell the difference in some situations where PCA can't, and they don't need color. So we advocate that we supplement the use of PCA plots with the use of RLE plots. So I'll just flash up my acknowledgements, and thanks to everybody, and I'm happy to take any questions. Although I basically used up the question time. Sorry about that. I went a bit over.
Yeah, we have 5 minutes of break time, so I can take some questions.
Oh, really? Okay, yeah.
Yeah.
So the next talk starts at 30:30. Okay.
It's fine. Yeah. So the question from Stefan Pedersen is, great talk, Luke. Thanks. If dealing with a scenario where differences between biological groups are extreme, would you suggest to perform RLE within treatment groups? A realistic scenario along these lines might be ChIP-seq, where a ChIP target is cytoplasmic in one treatment and nuclear in another.
Yeah, so maybe I should, yeah, so RLE plots, they are not infinitely robust. At some point, the biology can be strong enough to start pushing them around. And so if you're dealing with a scenario where there's very, very large differences in biological conditions, then indeed it might be good to restrict attention to single biological groups. Although the problem with that is that you won't see technical variation that is that might exist between your different groups. And indeed, if the technical variation is completely confounded with your biological group, and so if you've got, say, group A and a technical effect just specific to group A, and group B that has a technical effect that's confounded with group B, then your ability to see that difference, well, it's not going to be there if you're just plotting a particular treatment group. So in other words, I'm saying that for a vast number of cases, RLE plots are robust enough to do the job of visualizing technical variation while ignoring biological variation. But there's a point where the biology becomes so extreme that RLE plots aren't even useful either. Although I should say we have done some simulation, we have pushed the simulation to a limit and to the limit, and we know that RLE plots are very robust, but indeed they will eventually break down. And in that case, I don't know, if you have suggestions on what to do in that case, I'm all ears, because it will be handy to know what to do.
Thank you. Looks like it's really interesting. It's an interesting question. There is another question. Somebody have missed some part, and he's asking, how can we confirm whether the normalization method also discard the biological meaning the data might imply through RLE plot?
That's a great question. So that goes to the point of what we're saying, that in some sense, RLE plots are really, they're only useful for when it comes to visualizing technical variation. In some sense, because they're just, like, in some sense, just blind to biology, they're not useful for visualizing biology. So really, that's why we say supplement PCA plots with RLE plots. So PCA and RLE then have two different roles. At least this is how we use them. So when we see technical variation in data, we look at our PCA plot to assess clustering of biology, and we look at our RLE plot to assess whether we've removed technical variability. And you reach sort of success in our mind when you've got a nice looking RLE plot, because that suggests you've removed all the technical variation. And then you look over at your PCA plot, and if you see nice clustering of biology, that means you haven't also removed biology in your normalization of the data. Because that's one thing an RLE plot will not tell you. It will not tell you whether you have so-called thrown the baby out with the bathwater, because it doesn't see the biology. It won't tell you if you've removed it. You go over to your PCA plot to see whether you've still got your biology there.
Thank you. I think that makes sense. So the take-home question, take-home message is to use PCA and RLE plots together.
Indeed, indeed. We love them both.
Right. That's very interesting. Thank you.
Thank you. Thanks for the opportunity.
Yeah, due to the time limit, we have had to go to the next speaker.
Sure.
Thanks, Luke, for the very interesting talk. Yeah, I think a lot of people are still posting questions, so you can discuss in Slack later.